Thank you.
Could I send a link to Kelleher, or do you know if they got the link?
For the panelists?
Specifically, yeah.
Yeah, they all
you
should have
did.
been invited. Stephanie Chung, Kelleher, Cree Green, Bruning Graber, and Previs.
Excellent, excellent.
Right.
And let's see, I've got the stream link here. I will copy that.
Okay. And if you put it in the chat, then that'll be available to anybody who wants to send it to their friend or their colleagues. And then I'll set up.
Hopefully Melanie won't try to do multitask.
Yeah.
She needs to enjoy her vacation.
Yes, she does. Yes, she does. Okay. I think with 153 people signed up, Ben, things are going to get crazy, as you know.
Okay.
And I'll try to also monitor the chat for people's questions at the end, Owen.
Yes. So I'll, after my second slide, I'll tell them what the rules are. Basically, please submit a question via chat, and then one of the panelists will look at all the questions and pose that question to me. And hopefully it'll create a little discussion amongst the panelists if the topic is worthy of it. Well, needs more information.
Okay. Okay, so I'm gonna, Owen, I'm gonna tweet the live link. So right now, anybody, after this, anybody can be able to see.
Okay, so I'm live.
You're, yeah, in just like a minute here.
Okay.
I'm also gonna test it before I send it. Yep, looks great. Thanks, Ben.
You're welcome.
Are you going to say something before I talk? I'll just, okay.
I don't need to. I don't need
All right,
to.
good.
Because we're lots of people know you, but there might be a few new students who don't. If you would just
Okay.
give a
I'll
little
give a
bit.
background.
Yeah, and really, what MMPC has really been such a great resource, and some
Yes.
people still might not know about it. All right, here we go. I also think the timing of this during the day, finally the people on the West Coast can have a reasonable time
Right.
for a seminar for a change instead
Well,
of
and it's not right at noon either, so noon seminars don't necessarily hit up with those too. So.
Yeah, no, a noon seminar is great in California, so
yeah,
we'll have people zoom in.
Uh-oh, David raised his hand already.
Good. Hi, Dr. Wasserman. So good to see you. Not see you. So, and I should let you know that my Twitter filled up this last week because I was voted as vice president of TOS.
Oh, great.
To be president of the Obesity Society in its 40th year in 2 years.
Awesome.
so lots of stuff going on, and the Obesity Society meeting is next month, and it should be really good.
Good.
Did you see the survey that went out about COVID's impact on NIH grants? Did you happen to get copied on that? There's a very nice research project going with when you answer the survey consent form.
Right.
Asking about which issues were that had the most impact on NIH grants, and animal work versus human work, and
Right.
I'm really interested to see the results.
Hyperplasia, and we couldn't confidently rule out late cell tumors. So then we shifted into kind of a
So
monitoring.
I am now hearing a live stream
We're getting a lot of MRIs to make sure there wasn't any type of.
somewhere.
Is that Stephanie's audio? It's Stephanie.
No, is
It
it?
was Stephanie's audio.
There she is. Now you're muted, but we can hear your computer.
I'm sorry. I'm doing 4 things at one time as usual. Let me,
Yeah.
let me leave one meeting so I can come
Yeah,
to the next one.
Absolutely.
It's like as many devices as you can hold in your pockets.
Yes.
And I think the students online will completely understand that.
I'm
Yes.
sorry, I turned it off. How are you guys? Good to see you.
Good to see you. We normally would have had the tracer course last week, but
Yeah.
well.
Yeah, I even did my hair for it.
Okay.
These days.
Oh, yeah, it hasn't looked like this since probably I saw you last year. A tangled mess usually.
Yeah. No, mine is just sticking out due to stress, right?
So,
Yeah.
how many people are? Oh, we already have 28. Okay.
We're expecting over 100.
And they will all drop in here in the next 3 minutes. So we're being patient to let everybody catch up with us.
Okay, that's cool.
Yeah, the live stream is in the chat. If anybody listening to us now wants to forward that to anyone who didn't register. We have fall weather. We have football weather now. How about you guys? We were supposed to play Vanderbilt, but something terrible happened, and somehow that game got canceled.
Somehow we're not in the middle of a pandemic. No.
I don't know the scuttlebutt on that one, but my feeling is for this year, Vanderbilt's lucky they can field a team.
Yeah, well, and I think you beat us last year. I'm not quite sure. I'll get corrected, I'm sure, if I'm wrong.
We have pretty good weather too. It actually warmed up to even 70s, I think.
Well, it's almost 80 today, but it was beautiful. It was crisp 2 days ago, just perfect. It was about 39, and got out on my bike and it got to about 40, and it was, just chilly, but once you get going, you're fine, right? So
yeah, well, you're going to have a couple people come to Nashville, aren't you?
Yes.
For the debates.
Yes, it'll be busy. They've already started cordoning off the roads and stuff. So,
yeah, yep.
I
Please
know
stay
my
inside.
ride home is gonna be a little crazy. So.
Yeah. Take the alternate route.
Yeah.
Belmont's literally across the street from Vanderbilt, so that's the problem.
Yeah, it's going to be be interesting.
Well, I think we should look ahead. Next year cannot possibly be as bad as this year.
No, you can't say that.
Well, why not? It can only get worse. I mean, it can't get any worse.
We're just trying to stay flexible here in Columbia, Missouri. Whatever you throw at us,
We'll adapt.
What do we need to do next? And our leadership has been pretty good at keeping us up to date.
Yeah.
Football, as I said, the football schedule keeps changing.
Yeah. Well, I think it's a nice distraction, even though it is maybe not
It's
the best
Henry.
distraction.
Yeah, well, and it turns out I can buy a cardboard cutout. And I
Ah.
actually would like a picture of myself as a cardboard cutout
Okay.
in the stand. Good afternoon, Dr. Brunegraver.
Good
Morning.
afternoon, all of you.
Hi, Dr.
Hey.
Graver.
Okay. It is now 1:59, so one more minute and we'll start. And Ben, I'm going to turn off my microphone and my video till Owen is ready for us.
Henry, you probably need to turn off your microphone, and then you can turn it on at the end of the talk. Okay, welcome everyone to part 2 of Tracing Hepatic Glucose and Glycogen Metabolism Metabolic Disease. My name's Owen McGinnis. I'm a professor in the Department of Molecular Physiology and Biophysics at the Vanderbilt University School of Medicine and Basic Sciences. I also am Associate Director of the Vanderbilt Mouse Metabolic Phenotyping Center, which is a center as part of a big consortium of a number of centers providing services to investigators all over the world to study metabolic disease in mice and other rodent models. This is a great resource for everyone, and I welcome to go into the National MMPC website and see what services are available. Excuse me. Today, the format of this talk is that we're going to rely upon people to use the chat feature to post their questions. And then we have a distinguished group of panelists that'll help field those questions and post those questions to me and to the group as a whole. And our panelists are Henry Brunegraver at Case Western University, Steve Previs at Merck Pharmaceuticals, Elizabeth Parks at University of Missouri, Joanne Kelleher at MIT, Stephanie Chung at the NIH, the Endocrine Division, and Melanie Cree-Green at University of Colorado. I should remind you, this is part 2 of a 3-part series of these isotope traces in metabolic research. The third part will be done by Matt Merritt on November 17th. And the purpose of this talk is really to highlight a program that we've been running for the past 13 years entitled Isotope Traces in Metabolic Research. That was originally scheduled to be held last week, and we invite about 70 to 80 people come and sit for a week in a hotel and talk about science and tracers. We teach about the basics all the way to the advanced technology study metabolic research and applying isotope tracers. We tentatively have moved a new schedule till April 16th, and people from all over the world, graduate students all the way to faculty members, come to this, and it's a great opportunity one-on-one to talk about your science and the applications of tracers. And before I get to my talk, I want to let you know I had some disclosures. I have relationships with AstraZeneca, Calico, and Janssen. This particular talk and some of the data I'm going to talk about is actually from a recent publication that came out in Nature Medicine using a compound developed by AstraZeneca, which is a combined GLP-1 glucagon agonist. And really the purpose of this talk is not to focus on the drug, but to focus on the toolkits we have that can focus on regulation of hepatic metabolism and metabolic disease. So I'm going to give you a little outline of my talk, and I talk about some overview of some glucose and gluconeogenic substrate in fed and fasted states. I'm going to talk a little bit about how obesity affects insulin action and hepatic nutrient flux. I'm going to talk about GLP-1 and glucagon as it relates to nutrient homeostasis with a liver-centric view of this, really to help understand this targeting approach, and then look at the effect of combining targeting of GLP-1 glucagon signaling and hepatic metabolism. So let's start off with a really simplistic approach that we think about. We have these organs in our body. In the fasting state, the liver is the major source of glucose, supplying majority of the glucose released by the liver in the adult human is taken up by the brain, about 60% of it. In the mouse, it's a little bit less, 15 to 20%. And the amount of glucose is then taken up by peripheral tissues, primarily muscle and others. But that muscle primarily doesn't oxidize most of that carbon. That carbon scale is recycled as either amino acids or lactate back to the liver through a process of gluconeogenesis, which is used to supply the glucose. And we have this natural feudal cycle. We also have glycogen stores that also supply this carbon skeleton to support glucose production. But remember that in synthesizing something, you need energy to support that. And we have carbon skeleton in adipose tissue, primarily as fatty acids and glycerol, that are delivered to liver and peripheral tissues. The glycerol is used as a gluconeogenic substrate to support glucose production by the liver. The fatty acids are either oxidized by skeletal muscle and other tissues or sent to the liver, where about 30% is oxidized, and the remainder are repackaged, esterified, and sent out as VLDL, and we have this natural feudal cycle here. So there's a natural cycling of lipids and carbohydrates even in the fasting state. In the fed state, our carbon is coming from the intestine. In the case of glucose, the liver switches to a big glucose consumer of glucose as opposed to a producer. The brain still takes up about the same amount of glucose, even though the endocrine status is very different. Adipose tissue starts taking up significant quantities of glucose, and muscle takes up a large fraction of this glucose. The majority of that glucose is stored as glycogen, but there is also recycling of some of this carbon back to the liver for gluconeogenesis. So this process of recycling is ongoing both in the fed and fasted state. And from the adipose tissue, yes, we are releasing less glycerol and less free fatty acids, but that esterification process still is ongoing that happens in the liver. The muscle will see less free fatty acids, and that's why it can take up it's taking up so much glucose. Now, these metabolic changes that happen occur because there are endocrine changes that accompany this. In the fasting state, we'll have relatively low insulin environment, high glucagon tone. And if we look at it from a liver-centric point of view, that low insulin environment facilitates glucose production, and the high glucagon environment also facilitates glucose production. And this glucagon environment facilitates the carbon movement through the liver to make glucose, as well as supporting fatty acid oxidation in the liver. In the fad state, insulin levels are high relative to glucagon. And in this state, this high insulin environment is facilitating glucose disposal, acting as a brake on adipose tissue lipolysis, and therefore subserving a process of disposing of this exogenous glucose by the liver. Notice it's a consumer now, and stimulating glucose disposal, allowing us to have a normal glucose homeostasis. And while this slide depicts that we live in both the fed and the fasted states, the reality is that most people live more in the feasting phase of life than in the fasting phase. Because even when you're eating and then go to bed at night, you're still absorbing what you ate after your late evening snack. So that fast is not as long as you think. But that means that the biology of this control system is always working. We always transition from feeding to fasting, or relative feasting, relative fasting. So metabolic disease is really not only a problem of transition between feasting and fasting, but in having the capacity to rapidly switch between these processes. Obesity is really a state of metabolic dysregulation that compromises its ability to transition between metabolic states, and as a result leads to diseases. And if we think purely from a liver-centric point of view, this is presented as lipid accumulation or non-hepatic alkalosteatosis that can progress forward to liver disease. And this dysregulation is quite robust. And the reason why it happens because obesity creates a state of hormone resistance, and the classical one is insulin resistance. And the way we quantify insulin resistance is we form a hyperinsulin euglycemic clamp. So we infuse insulin in a fasted state, And then we try to hold the blood sugar constant by giving exogenous glucose, and you'll see the exogenous glucose infusion rate goes up over time. So the amount of glucose we have to give tells us how well this insulin is doing in supporting glucose homeostasis. So you can imagine that you could give different doses of insulin and get different glucose requirements. If you couple this with tracer methods, you can quantify how much glucose is moving in and out of the body. And so if we plot dose of insulin, the insulin concentration versus glucose disposal, how much glucose is used by the body, in a chow-fed animal, what we see is a nice insulin dose response curve, that as we raise insulin, whole body glucose disposal goes up. In an insulin resistant state or high fat fed state, the dynamic range of insulin is dramatically blunted. So your ability to get maximal glucose disposal is impaired. In addition, you have to have much higher insulins to get physiologic glucose disposal rates, which is the range that I'm highlighting here is that we work in. And so if we think about this, we live in the fasting state in these two individuals with very similar glucose disposal rates, but they have to have very different insulin environment. In the fasting state, the obese individual has to make a lot more insulin to get rid of that whole body glucose disposal, even in the fasting state. And so if you take a pick and just measure how much glucose is made by the liver in the fasting state, low fat and high fat, what you'll see per kilogram body weight that the liver is actually making less glucose. But if you look at the contribution of gluconeogenesis in the mouse, it's about 70–75% of the glucose coming out of the liver is from gluconeogenesis. But wait a minute here. When we look at this data, this is milligram per kilogram body weight. Well, when you're overweight, you weigh more. So if you just ask how much glucose is being produced by the liver and express it just in total mass, in fact, the amount of glucose made by the liver is actually higher in the mouse in a high-fat fed animal in terms of milligrams per minute. So that the increase in glucose production we're seeing here is due to combined effects of increasing gluconeogenesis and increasing glycogen breakdown. And there's this driver of glucose production that we want to talk about a little bit today, and this ability to turn off glucose production in a state of insulin resistance. So if we did an experiment and we took, these are basal insulin levels in a low-fat fed animal and a high-fat fed animal, which you can see they're much higher in the high-fat animal. And then let's infuse exogenous insulin to raise insulin levels in both groups. The first thing you'll notice is that the obese animals have higher insulin levels even in the resting state and during the clamp. So this is the challenge, to look at this data, understand, so what does insulin resistance look at when I start with different hormone environments? So the way to look at this is actually to plot the insulin levels you get versus glucose production. And so here, if we look at the chow-fed animal, you can see the liver's making 20 mg per kg per minute. And when we raise insulin, the liver stops making glucose. The obese animal is making maybe a little bit less glucose in this example, but can respond to insulin, but requires a lot more insulin to produce the same biological effect. And notice it doesn't completely suppress with this insulin infusion rate. So clearly this animal is insulin resistant because it has to live on a different part of the insulin dose response curve, but it can respond to insulin. So therapeutics have come along to try to correct this insulin resistance in the liver. And I want to just talk about a class of drugs that it's been around. It's called GLP-1 agonists. And if you want to improve insulin action in the liver, the best way to do it is actually lose a little bit of weight. And what these drugs tend to do is help you decrease caloric intake or cause satiety. The other thing they do is they help you secrete insulin. They stimulate glucose-stimulated insulin secretion, so that maintains glucose availability. So as your glucose control becomes more and more difficult as you age and become insulin resistant and progress from insulin resistance to diabetes, this can protect your beta cell and augment insulin secretion and try to maintain glucose homeostasis. Remember what I told you before, that this transition between fat and fasting states is so important and a real challenge for us. So by delaying gastric entering, of slowing that food entry rate, it puts less stress on our system in facilitating glucose homeostasis and maintaining glucose control. It also can enhance the slow down the rate of entry of triglycerides from diets, also therefore really limiting the stress in our system. The other thing it does is it lowers glucagon secretion in the fed state. And so therefore you might think in that case that that would suppress glucose production and facilitate glucose disposal. But life's a little more complicated than that, as we'll see as I get into talk. So let's think about glucagon for the moment. So we know that glucagon in isolation is a very potent effects on the liver. It's a potent stimulator of hepatic glycogenolysis, a very potent stimulator gluconeogenesis, both in raising the capacity of the liver to synthesize carbon from gluconeogenic precursors, as well as stimulating amino acid uptake by the liver, those precursors that are used to synthesize that carbon. And also, as I pointed out early on, these processes of gluconeogenesis is energy requiring, so it is complemented by an increase in fat oxidation by the liver. And at higher doses, glucagon can cause weight loss, which will complement the GLP-1 effects, and also can increase energy expenditure in some settings. So the question we came to the approach we were thinking about was, so we have these drugs, they've been on the board, and we've got pretty decent clinical trial data that says if we give GLP-1 with a little bit of glucagon, glucose control is better. Insulin action seems to be a little bit better, but really, if glucagon is working really well, wouldn't it impair glucose disposal by the liver? Would it force the peripheral tissues to have to do more work because the liver's not doing its part in disposing of that exogenous glucose? So the focus of this study was, could we use tracer approaches to ask whether the role of the liver in disposing of glucose in the fed state is compromised by adding glucagon on the background of GLP-1 agonism. So the experiments we did were we used black 6 mice that were fed a high-fat diet, and they were treated daily with these drugs of either liraglutide, which is our GLP-1 agonist, or codaglutide, which is a combined GLP-1 glucagon agonist. And in this paper, we also gave a pure glucagon agonist as well, which I'm not going to talk about. And this was given for 7 days. So because we were focused on in the fad state, what we did was we shifted the light-dark cycle so the experiments were done and the treatments were done at a time just before the animals would normally eat. So we shifted the light-dark cycle so that dark was midnight, but it was actually at noon for these animals, and we gave our drugs at 10 in the morning. So we timed the treatment to match when they were eating food. And they were randomized for body composition and fasting glucose before we did the dietary manipulations. And to test the capacity of the liver to store carbon and metabolize that carbon, we took these 5-hour fasted mice, but remember, these are 5-hour fasted late in their, quote, light cycle. So actually it's a longer fast because they haven't been eating much, okay, on their 7th day of treatment. So what we did was we did hyperglycemic clamps. The what we used was one of the toolkits that are well known in the Vanderbilt metabolic MAPC is our chronically catheterized conscious mice. These mice have catheters put in a jugular vein and a carotid artery in 5 to 7 days before an experiment. Then the animals put in the cage, so they have free access, they're completely conscious, so we have complete vascular access. So now we can infuse tracers of, in this case, we're giving deuterated water and 6,6-dideutyrated glucose and red blood cells to replace the blood that we have to take during the study. And then we're going to take samples from these animals over time and look at the labeling of glucose glycogen, and the glyceride and the palmitate and the products of synthetic reactions ongoing in the liver. So the way this design is we give the 6,6-glucose and deuterated water in advance, allow equilibration of these tracers, and then we measure baseline glucose flux here, and then we perform a hyperglycemic clamp at 280 milligrams per deciliter for 2 hours. Now let's look about why we chose these tracers. The advantage of the 6,6-glucose is that it's going to tell us how glucose is moving throughout the body. So it's going to be our measure of whole body glucose flux. The deuterated water is going to be incorporated in synthetic reactions that occur within the liver, since we're looking at the liver today. Our treatments are 3, liraglutide and codaglutide, and the vehicle, just to remind you. And so when we give deuterated water is going to be incorporated into metabolic intermediates in the citric acid cycle, as well as the glycolytic pathway on the way to synthesizing glucose. That deuterium will incorporate in this place here, as well as just before glucose 6-phosphate. So therefore, any glycogen that comes out into G6P, because there's a rapid equilibrium here, will actually pick up this deuterium here. And because the deuterium is incorporated at different steps, it will incorporate in different places within the glucose molecule. And so what we can look at is measure in the glucose molecule using GCMS where the label is. And we could look at the label in which position of that glucose molecule it's located in. So this will be the liver when it's making glucose. And this is all carbon flowing through the gluconeogenic pathway. Notice that the carbon is coming either from glycerol, from lipolysis, or from lactated amino acids that are coming through the pyruvate pathway, enter the TCA cycle through this mechanism. Now, in the fed state, carbon flow is different. Here now we have hyperglycemia. Glucose carbon is going to enter the G6P pool, where we're infusing 6,6-glucose. So the glucose that's labeled from our infusion will enter the G6P pool, and it has 2 fates. It could enter the glycolytic pathway, or it can go into glycogen and be deposited as glycogen. If we look at the end of the study, we can look at the labeling pattern in glycogen. Now remember, glycogen can be synthesized from glucose into glycogen. We call that direct glycogen synthesis. But remember, lactate and glycerol all entering the pathway down here. This gluconeogenic pathway will still be on despite the fact that the liver is in uptake mode. This carbon will be diverted into glycogen. We call that indirect glycogen synthesis. And so when you look at glycogen synthesis in the fed state, it is comprised of both direct glycogen synthesis and indirect glycogen synthesis from carbon precursors. But at the same time, that glucose carbon can be incorporated into the triglycerides that are being synthesized in the liver. And they're coming from 2 portions. The glycerol backbone forming the glyceride is coming through diacylglycerol, and this is going to get incorporated deuterium in here. So this deuterium will be incorporated into the triglyceride molecule in the glycerol portion of the triglyceride. The triglyceride portion also will have fatty acid molecules that will be labeled. Those labels will come from acetyl-CoA that will be labeled in the de novo synthesis of free fatty acids. So this triglyceride molecule will have free fatty acids that will be called de novo, or newly synthesized fatty acids. They will be labeled. And free fatty acids that came from peripheral tissues that got re-esterified and formed a triglyceride molecule. And in most situations, the exogenous free fatty acids are the dominant source of the free fatty acid carbon that's used in acylation and forming the triglycerides. But because we raise glucose levels, we will actually force carbon up through this synthetic pathway as well. And because we can look at the labeling pattern in using GCMS, we can actually look at how this triglyceride, what carbon skeletons were used to make this triglyceride molecule. So in this experiment, we gave these drugs. In this vehicle group, basically body weight did not change. They were already overweight. They were about 45 grams in size. And we got a mild weight loss with both drugs, a very mild loss, about 3 to 4 grams of body weight, a very mild weight over 6 days. And during this window of time out here, this is where we perform the hyperglycemic clamps. When we take it measure those fasting glucose levels, what you can see is that, remember, these animals are being measured at glucose. It's just before they eat. So the glucose levels are actually quite low relative to what many of you might be used to looking at a high-fat fed animal at 6 or 7 in the morning, where the blood sugars would be 130, 140 milligrams per deciliter. Remember, this is late in their day because we shift the circadian cycle. So the glucose levels are absolutely lower because they're more fasted. Notice that the combined GLP-1 agonism actually lowered glucose levels, and we'll talk about that in a minute, but it was not associated with a fall in insulin levels, whereas the liraglutide had lower insulin levels. So the drug has been on board for about 2 hours, but we don't have hyperglycemia, so we don't have a potent stimulation to the GLP-1 dependent insulin secretion. If we look at basal hepatic glucose production, what you'll see is that the presence of the glucagon agonist actually raised basal glucose production, reflecting that 2 hours before we gave a drug that has glucagon agonism activity and stimulated glucose production. All right. Liraglutide, there was no change in glucose production. And if we look at the rate of glycogenolysis, it was not affected in this study. And gluconeogenesis was the primary reason why hepatic glucose production was up in the presence of this glucagon agonism. Yet glucose levels were not worsened by this. So then we performed hyperglycemic clamps. So we clamped them all at around 300 milligrams per deciliter and held it there for 2 hours. And not surprising, when you raise glucose, you make a lot of insulin. Remember, the basal insulin levels are very low in this setting relative to what we see. So this is a high insulin tone environment. And you got very similar insulin levels in all 3 groups. The codotype get a slightly higher insulin secretion, probably because their basal glucose levels were running a little bit lower. So there was a greater stimulation of insulin secretion possibly present with this. But let's look at what the liver is doing. Not surprising. As soon as we raised the glucose level, the liver stopped making glucose in all 3 groups. The liver stopped making glucose. And if you look at gluconeogenesis, that is the carbon coming out of the liver, it is completely suppressed. And glycogenolysis is completely suppressed. So while the liver was making lots of glucose in the basal state, once we raised glucose levels, the liver was no longer a source of glucose carbon to glucose production. Now, just to remind you, this is a fed state, so therefore glucose is going to go into glycogen. All right, so we're going to look at this glycogen pool here. So glucose is going to come in. It's either directly go into this glycogen pool, or glycogen is going to be synthesized via this indirect pathway to make glycogen. So the first thing you do is measure total liver glycogen, and you see it's about 30 milligrams per gram. It tended to be a little bit lower in the glutide group, but this doesn't tell you the story, because remember, this is the terminal liver glycogen at the end of the clamp. So this glycogen represents the core glycogen before any synthesis occurred, as well as any newly synthesized glycogen that was deposited during the hyperglycemic clamp. The beauty of the tracer method is that any core glycogen that was there before the study started will not be labeled. It will be unlabeled. Any newly accrued glycogen will have label in it. So when you take the glycogen and degrade it, you can look and ask how much of that glycogen was newly synthesized versus how much was there before. And therefore, we can calculate what we call the core glycogen, the glycogen before we did the hyperglycemic clamp. And consistent with what we saw, what we'd expect with the glucagon agonist at present, glycogen levels were a little bit lower with glycotide. So you might say, well, what happened to glycogen synthesis? Well, think about it. It's really the difference between this bar and this bar. And if you calculate it, you'll see that the total glycogen synthesized was about 20 to 25 milligrams per gram liver. Very similar between the groups. So clearly having the glucagon agonism present in the presence of GLP-1 agonists did not compromise glycogen deposition. And if you look at the direct glycogen synthetic rate, about 70% of that glycogen was synthesized by the direct pathway, which means about 30% was via the indirect pathway. And that's quite common in many model systems. It's about 50% depending on it. But remember, because we did a hyperglycemic clamp with peripheral glucose infusion, that will preferentially augment direct glycogen synthesis. In a meal, when you eat a meal, it can augment indirect glycogen synthesis because all those amino acids that are in the meal will actually become carbon skeletons for gluconeogenesis that actually fill glycogen via the indirect pathway. So you can get a greater glycogen synthesis because when you actually eat a meal, you're actually not just getting glucose, you're getting other carbon skeletons. In addition, when you eat a meal, there's a portal signal effect that when you give glucose via the portal vein, or you give something enterally, it further enhances glycogen deposition as well. So this is peripheral hyperglycemia augmenting glycogen synthesis. So remember, the liver is in uptake mode. It could take this glucose and make glucose, but it also could make de novo lipid or newly synthesized palmitate. And when you look at total palmitate in the liver, now it represents palmitate's probably about 20% of the total lipid in the liver of the fatty acids, depending on the diet you're on. It's a little bit low with the coraglutide, which I'll talk about a little bit why it might be a little lower. But notice both liraglutide and coraglutide both tended to give you less de novo lipogenesis. And it's also a small number relative to the total palmitate in there. So this newly synthesized palmitate is a small fraction of the total amount of lipid that was esterified in the liver. And it's most obvious when we look at the next slide. If we look at total glyceride in the liver, we see a total decrease in liver glyceride reflecting the lower lipid content in the liver. But remember that glyceride backbone also is being newly synthesized as new fatty acids are being esterified to form the triglyceride. And notice that this represents about 20% of that glycerol in the glyceride has been resynthesized in that 2 hours. So this pool is rapidly turning over, churning out lipids and packaging it and sending out of the liver, some of which is staying. We're only looking at what stayed in the liver. And what you can see is that this newly synthesized glyceride was also low. So the liver had less palmitate and less glyceride, but similar DNL. So likely what's happening is that we are actually seeing less total esterification that's actually occurring in the presence of liraglutide. Now, why might that be? The glyceride flux rates are very similar between these two drugs, but remember what glucagon does. It also stimulates fat oxidation in the liver. And so maybe some of the fat that's being delivered to the liver is being diverted to fat oxidation, is not therefore available for glyceride formation and esterification. So when we look at static numbers of total glyceride, you really lose the whole picture. But by adding in tracers, you begin to learn how dynamic these pools are and where those carbon skeletons are coming from. So if we go back to this idea of feasting and fasting, in the fasting state, the liver is making glucose, and we'll use a number of 10 milligrams per kilogram per minute per unit time. A lot of that carbon is derived from glycogenolysis and gluconeogenesis from this recycling of carbon. In this feasting state with hyperglycemia, we converted the liver to a big glucose consumer, 5 milligrams per kilogram per minute. That's a dramatic shift. So this is a large reapportionment of glucose carbon. But remember, there's still a lot of carbon being taken up by peripheral tissues in adipose tissue and muscle that then have to either oxidize, this primarily in skeletal muscle, and adipose tissue esterify that glucose, or de novo lipogenesis. In the context of 2 hours, the total amount of de novo lipogenesis would be relatively small in adipose tissue. But if you think about our life cycle of living primarily in the feasting state within a 24-hour state and a relatively high carbohydrate diet that we live on, this DNL is actually pretty significant in the adipose tissue and actually can be significant in the liver, especially in settings of obesity and de novo lipogenesis, insulin resistant states. So when we think about this, biology, we have to realize that all this biology is complicated. And really what we hope in this, if you attend this course that we're offering, is it was funded through training an R25 called DK11362. These programs that were supported by the metabolic micro mouse program that supported this course, and now also supported some of the studies that were developing some of this technology we applied to study metabolic systems. And then the Mouse Metabolic Phenotyping Center, which provides the toolkits and access to make it this really sophisticated technology available to the larger diabetes community and the diabetes centers as a whole. And now I want to remind you that there's part 3 of this talk, and Matt Merritt's going to talk about central metabolism as a target for pioglitazone action. And remind you that this webinar, as well as all the other webinars, will be recorded and shared on the website. And so just to remind you, if you have any questions, post a chat and we'll get to discuss. I intended to make this talk short to give us a great opportunity to allow discussion around these topics. Thank you very much.
I'm gonna clap for you. Really beautiful, elegant work. I really wanted to start off by asking a question about, in terms of that, this was a 2-hour clamp in mice, is that right?
That's correct.
At a single insulin dose. And so the idea then, the bottom line of how much glycogen is actually turning over and being replaced from either source, from blood glucose or from gluconeogenesis during that time is what percent? I mean, it's a huge percent, right?
So in that 2-hour time, the glycogen synthetic rate is about 5 milligrams per kilogram per minute, right?
Yeah.
In a hyperglycemic state. If you are fasting an animal, right, during that 2-winter time, the liver would be making about 10 mg per kg per minute. About 70 to 75% would be from gluconeogenesis, and about by that time 20 to 25, because the glycogen stores would slowly be depleted over time.
Yeah.
So that gluconeogenic pathway is still on, and I think a lot of people forget that. And it's on all the time, and actually it's a good thing, because every time you eat a meal, you have protein in it and amino acids. That nitrogen has to be disposed of by the liver, and that carbon skeleton has to go somewhere. That gluconeogenic pathway, by having it on all the time, both in the fat and fasted states, allows that flexibility of the liver to always be working. It's always working. It's never quiet. And we forget that that oxidative capacity of the liver, to maintain that oxidative capacity, really is important, not only in the fasted state, but in the fed state.
Yeah, yeah. Thank you. We do. Go ahead, Stephanie. Are you?
Okay. I was trying to see if there was a question. I didn't want to take somebody else's time, but while we're waiting, oh, and that was a really beautiful talk, and I really appreciate it. We do stable isotope studies looking at rates of gluconeogenesis and turnover in kids and humans. And so I guess I wanted to ask, with this particular drug, do you have any sister studies that are going on, or is this really still in the preclinical model in terms of assisting, for patients with diabetes?
So the answer is actually there are phase 1 trials already being done with this drug. And I'm not privy to all that data, but I know that it's been working on, and at least the initial data looks promising. I think in this field of, it's sort of counterintuitive. You always view the glucagon and insulin as a yin and yang. When insulin's present, you want to turn off glucagon.
Right.
And the problem is in metabolic disease, where nutrient excess is really high and nutrient availability is really high and lipid availability is high, you can make lots of insulin, but insulin doesn't support the oxidative component of the liver. And because the carbon flow is so high, if you don't have some glucagon tone or something like it to support that oxidative machinery, you'll put yourself in a position where you have more carbon than you can handle. And this creates what we call oxidative stress, volume overload for the liver, and it has to figure out where to put that carbon skeleton when it can't oxidize it.
Excellent. Have you done similar studies with metformin and looking at metformin as one of the comparators with liraglutide?
I have not. We have not, using this model, we have not done that.
Okay.
Hey, Owen.
Yeah.
Hello?
Oh, Steve. Yeah.
Hey, Steve. How are you doing?
Good.
So thanks for the presentation. Something that comes to mind, actually, you guys did, I think, one of the very early studies on this with Erwin Kurland,
Yes.
where you did not clamp, right? You did just a glucose challenge. And I know you guys at Vanderbilt have done quite a bit of that recently with propionate and labeled water, and the guys in Australia have done a lot using these rather simple, methods. As you guys are looking at the literature, so there's obviously nothing wrong with putting the catheters, doing the clamps and so forth. But as you look through the literature, how many examples do you find where, like you guys said, you could unmask a phenotype using the challenge test that you couldn't really pick up with a clamp? And, what's the trade-offs between the clamping versus non-clamping and throwing tracers in there?
Yeah. So that's a great question, Stephen. And really, you developed a lot of technology where you can use deuterated water to trace metabolic pathways in general, not just gluconeogenesis and a clamp technology. Really, you can use it for lipid synthesis. You can look at for DNA turnover and stuff. So the advantage of a loading study is that you can push a system and ask, is it capable of shifting between one metabolic pathway and another metabolic pathway? Then the positive is it tells you about the dynamic resiliency of that system, how well that system can adapt and transition between fed and fasted state. What it doesn't tell you is quantitatively how important one system is in another, because you have too many other variables you're not controlling. So the advantage of the clamp is you're controlling known variables that impact your variables. So you can then quantitatively ask whether one variable, let's say glucose or insulin, is the driver, a quantitative driver of process. So actually those approaches complement one another. There isn't one that's better or worse. We call the clamp a gold standard because it can be quantitative about one variable of insulin action. But the other practical problem is that not everyone can do clamps. But it's sort of it's the tertiary experiment that sometimes allows you to feel comfortable of knowing how that drug works.
Yeah, no, I agree with you. I think the use of the word complementary is really the critical thing, right? One is not really better than the other. It's context dependent, and they inform on different aspects. I was just curious to see, like, in the literature now, do you see, for every clamp study that's published, you have one using a non-clamp method, or is it really people still think really one way or the other?
I think the challenge with the isotope methods is there's an intellectual barrier to understanding how to interpret the data.
Yeah.
And I think that's the problem. And so while there are some analytical hurdles that one has to overcome, a lot of those have been conquered because now we have core labs that actually do the analysis for you. So the challenge really is designing the best experiment for your question, which is a real problem because most people don't design the experiment around the question they're really addressing.
Yeah.
And then data interpretation. And a lot of people get in trouble with misinterpreting the data because they didn't really design the experiment well enough or didn't understand the biology well enough.
Yeah. Thank you.
I can't hear you, Elizabeth.
Sorry, we've got a lot of questions coming in. And the first one is, did you measure serum lipid levels as well? HDL in the treatments at all? Is that in the paper?
Yes, that's all in the paper.
Okay, and I put the PDF of the paper in the chat if people are interested in it. A second quick one is we are studying, from Catherine Ray, we are studying patients who have long chain fatty acid oxidation disorders and can't break down fats. My question is, would the glucagon stored in the liver turn into liver fat and build up over time? And I think she means glycogen, maybe.
Yeah, I think that's what she meant. So glycogen is a readily mobilizable fuel. So as long as they can get it out of the liver, it's not going to be necessarily diverted down to lipid synthesis in a setting. In most glycogenolytic states, carbon is moving out of the liver. It can go down the glycolytic pathway and down to pyruvate and go out as lactate. I wouldn't expect DNL to be significant in that particular state. Now, I guess one could envision in fatty acid oxidation deficits where you actually may have significant amount of carbohydrate oxidation ongoing already in the liver. It's possible that you could divert some of it, but I think it's highly unlikely.
Yeah, yeah. There is a question about using tracers in human subjects, and we have a number of people online who on the panel who do that. So all of these methods are possible in humans.
Yes.
Can you comment on how delivery of insulin via the portal or peripheral effects our interpretation of the contribution of glycogen synthesis and GNG during clamp conditions? I can repeat that if you'd like.
All right, so I understand. So in this experiment, we didn't actually infuse any insulin. So the pancreas was making its own insulin. So this insulin was delivered into the portal vein. So it's a physiologic delivery of insulin. So this is not an infusion. It is true that if you give in a clamp setting, if you give a peripheral infusion of insulin, let's just say the insulin levels go from 0.5 nanogram per mil up to say 2 nanograms per mil. The level in the portal vein is actually slightly lower than the arterial level because some of the insulin gets metabolized and goes through the GI tract. So actually so the insulin tone to the liver is not as high as you think it is based on peripheral insulin levels. If you had those same peripheral insulin levels, but the pancreas was making the insulin levels in the portal vein would be about 3 times higher. And so when you look at peripheral insulin levels, when the pancreas is contributing to the insulin load is very different than with a clamp. And so insulin, peripheral insulin, is quite effective at stimulating liver glucose uptake, but portal's even better at the equivalent insulin concentration. But the complicating factor is with peripheral insulin, you suppress lipolysis more than with portal with the same dose of insulin. And so you get a complementary effect that as you suppress lipolysis, you make the liver look better at responding to insulin because you've lowered the fatty acid load to the liver. So when you think about insulin action in the liver and regulating glucose homeostasis, you can't forget that an important substrate that regulates how insulin works in the liver is actually fatty acid delivery itself. So in general, you can't think of fat and glucose and insulin as three separate buckets. They live in the same bucket. And so you always have to treat them as equivalents. And you can't ignore one. Oh, I'm studying glucose metabolism, so forget about lipid. It's not the best policy. I think this using the tracer approach of looking at lipid synthesis and glycogen synthesis in the same model, using one tracer actually is a way to realize that you don't have to get trapped into glucose. You can look at lipid as well in the same approach, using the same tracer approach.
I'll ask another question if someone else isn't going to. There's a comment by Clinton Bruce. Many years ago, it was shown that the low rate glucose infusions suppress endogenous glucose production, even when somatostatin was infused to inhibit insulin secretion. In light of this, how do you interpret clamp measures of hepatic insulin action where endogenous glucose production is likely being suppressed both by the glucose infusion itself required to maintain the euglycemia? And I know that's complex, but we're talking about effects of
I know this. I know the exact experiment you're talking about. So if you take a human and the liver is making about 2 milligrams per kilogram per minute of glucose, and if I would get a syringe and start pumping in 2 mg per kg per minute, in about 30 minutes, even less than that, the liver will stop making glucose, but the blood sugar will look completely unchanged. All right. But if you were really smart and you'd measured insulin every minute for about 30 minutes, you would have realized there was a little rise in insulin and a little fall in glucagon and a little rise in glucose, maybe 2 or 3 milligrams per deciliter. But the system, the beta cell, is so sensitive to glucose that 2 or 3 milligrams per deciliter was just enough to tweak the beta cell to tell the liver. And when you're looking at peripheral insulin levels, you may miss it. And so it says that the liver is very sensitive to glucose, but it's not the glucose infusion. It's the endocrine response to the glucose infusion. But you are correct that if I were to take you and give you somatostatin and clamp insulin and glucagon and everything flat and raise your blood sugar by only 5 or 10 milligrams per deciliter, the liver would stop making glucose by about 0.5 mg per kg per minute. So glucose itself suppresses hepatic glucose production. So that's why you need to do a good clamp. When you do a clamp, what you want to do is keep the glucose constant so that the glucose-dependent effects in the liver are not confounding the insulin-dependent effects in the liver.
Yeah.
It becomes more challenging when you have diabetic subjects who incoming glucoses are different than your control subjects. Then you're going to go, okay, what glucose do I clamp them at? So what people do in the clinical arena is they do an overnight insulin drip or modify the diet a little bit to try to get the glucoses in the morning very close to the same in both groups, so that when they go and actually clamp, there's not a big change in glucose in the clamp if they want to measure insulin action. But it is a real challenge when glycemia or incoming glycemia is really different between groups.
Yeah. I don't know if Stephanie, if you'd be willing to comment on how you choose what level to clamp at in different patient populations in your study, if you have diabetics versus healthy controls. What are you thinking about there?
Yeah, I think you're really trying to see what's as close to normal as possible, if that's possible. But you're gonna try to go within 10%. And you want to try to bring down the patients with diabetes and bring the glucose levels as close to normal as possible. So 100 milligrams per deciliter, you might have to sort of settle for somewhere around there rather than pushing it to the lower end of normal. It's not perfect, but at the same time, you have to really look at the physiology, and getting someone with diabetes perfect control overnight isn't necessarily the easiest thing. Unless you're sleeping by their bedside and doing it every 2 minutes.
Yeah. And so what you're talking about is infusing insulin overnight and glucose ahead of time to try and lower that diabetic's fasting glucose production. Correct. And then you do the clamp. And the question, and so this is the hard question I always have is, haven't you then just changed the clamp because you basically changed their metabolism overnight? How do you think about that?
That's a good question. I think it's a struggle. It's a trade-off between really going through the clamp and having differences in glucose levels induce differences in hepatic glucose production versus changing the milieu. And so my solution to that is not to do some clamps in certain cases and to look at the physiologic state and to use other techniques. But I don't think that there is one right answer. I think you need to understand the limitations and the assumptions when you do both, and importantly, treat your groups the same, so using the same study design and potentially using a paired model if you have an intervention, and making sure that you're comparing apples to apples within the same group.
Yeah, and when we're reviewing grants that have clamps in them, even mice and humans both, because there are so many ways to do this procedure and think about it, you just need to validate why you've chosen the way you're doing it and explain it to your reader for sure.
Correct.
Yeah, I think that it's always a challenge. It always goes back to what your hypothesis is. And you have to design the study to basically fight against your hypothesis, right? Correct. So you want to be absolutely sure that you're right. So you design the study basically so that if despite the best design, you come out that, my hypothesis was testable, then I think you feel comfortable with saying, my drug or my therapeutic or whatever is actually doing what I think it's doing, versus like, I may have to do the experiment 3 different ways to be absolutely sure. And human studies are so expensive. You got to do it right the first time.
Yeah. Do any of the other panelists want to weigh in or make a comment while I'm reading the chat here to see what other questions we might have? Give you just a couple seconds to think. Okay, there is a question from Eve, and this is really talking about the pancreas. It sounds as though the beta cell is very sick in type 2 diabetes, although now we are hearing that the alpha cell is also part of the problem. How do you think about that when you're looking at a glucagon agonist?
So let me start out with the problem of measuring glucagon. It's the nemesis of the field. It's a real challenge because the levels of glucagon in the fasting state are at the limits of most assays. And we know that glucagon, if you eat a mixed meal with lots of protein in it, glucagon actually goes up in a mixed meal. All right, and insulin goes up as well. I mean, but in diabetic individuals, if you give them a pure carbohydrate meal, what you'll see is that in a non-diabetic individual, insulin will go up and glucagon will tend to go down. In a diabetic individual, like glucagon will fail to fall, in some cases goes up. And we know that the failure of glucagon to fall has a huge impact on the ability of the liver to respond to insulin. But in this particular study that I talked about, we gave a drug that basically made sure there's plenty of insulin available. And so in a setting where insulin is in excess or in more than is needed, the glucagon doesn't act as a negative regulator of something, because the hyperglycemia and the hyperinsulinemia act as a counterbalance to that glucagon tone. But if I were to give glucagon in isolation with sort of the impaired beta cell function you have in a type 2 diabetics, that won't work out too well, because you need that extra insulin to counterbalance the glucagon that you're giving to support, say, fat oxidation in the liver. That's the problem. The diabetic individual, the impaired beta cell function limits their counterbalancing. And that is where they struggle. And that's also the clinical conundrum of this glucagon agonism, or drugs in that class. It doesn't have to be this particular drug, but drugs of that class really work best if you have enough beta cell function available, I think, in my opinion.
There is a question of, can stable isotope tracers be used in humans to measure nutrient flux in and during and post exercise? And there are thousands of papers where this technology has been used. There's even one of my top 10 of all papers of all time by Vernon Young, where he fed people and he measured substrate turnover, and then he exercised them, and then he fed them while he was exercising them at the same time, to look at food intake and insulin causing a disposal of nutrients at the same time exercise is causing a delivery of nutrients to the muscle. So there's excellent literature out there, and this technology has really taken us a long way to understanding the beneficial effects of exercise. Any other questions that you see, Stephanie or Steve, as we wrap up here? I'm looking. I know we haven't gotten all of them.
Yeah, I think Eve was asking or stating that she was unaware that glucagon was a problem, and that type 2 is a loss of beta cell. And I think I just want to hone in on what Owen has been telling us, is that it's really, you can't think of it in isolation. You know, the way I was taught it, it's really an insulin to glucagon ratio. And so there is a dysregulation in that insulin to glucagon ratio, and the drugs that he was talking about really is trying to right that balance a bit. And so, but measuring glucagon as well as intact GLP-1, you have to be very careful with the assays that you pick. I think we didn't talk about GLP-1 assays, but you want to look at whether you're looking at total or intact GLP-1 assays, and there's no standardization for those assays, but you can, there is good data out for several ones, and you just need to do your homework first and use the same assay and compare apples to apples throughout the experiment.
Yeah, great. Okay, Owen, you want to wrap up, and thank you so much.
Thank you, and remind everyone to attend the next one in about a month. Take care.
Thank you.
And all right, bye.
Bye.
Bye.